UMCCR's Council of Unmet Needs initiative, provide us an update on the VCCC Precision Oncology Program, as well as an overview of the University of Melbourne's Rapid Cancer Genomic Transcriptome Analysis platform. Could you please help me in welcoming Professor Sean Grimmond? Thank you very much.
Belinda, thank you very much. It's always traditional to thank the person who introduced you, but I really do think that you need to be congratulated there with that dash. Look, it's an absolute pleasure to be here this morning. Let's just see. Excellent. Sorry, I should say here this afternoon. And I'm very much looking forward to presenting to you a body of work here that really is the major focus of many of the laboratories within the University of Melbourne Centre for Cancer Research, but is really carried out in close collaboration with our partners throughout the VCCC Alliance and indeed nationally and internationally. And what I'm going to look to do with my talk today is describe to you how we've been working in recent years to build the key foundations we think are necessary on realising the dream of precision oncology, describing to you the battlefields that we've chosen when it comes to cancer to actively prosecute using precision oncology and investigating the value of thorough genomic testing on cancer patients so that we can improve diagnostics and improve clinical decision making when it comes to therapies. Just by way of introduction, UMCCR was established in 2017. We started moving in shortly after this magnificent building was finished. And it is a very broad church containing researchers working in basic translational and clinical research. But we're very much focused on seeing how we can create the tools that can be integrated into cancer care paths. And we're very much focused on seeing how genomics can provide insights into that. And the graphic I'm just showing you here is that is an example of the patient experience where we think about the path a patient will go, where we can see that genomics actually can contribute insights into a patient's disease from early diagnosis right through to when you might be thinking about combating their disease, through to how do we actually think about coming up with new approaches to improve outcomes. And then if you have a look within the UMCCR, if you haven't caught up recently with what we're up to, there are strong programs looking at the likes of prevention, to trying to understand how your genetic risk can contribute to your chance of contracting malignancy. We're very interested in using genomics for diagnostics. We're very keen to enable activities where we can allow people to test the clinical utility of genomics, whether it be in clinical trials or other studies. And then indeed, we're very keen to be actually focused more on how do we use genomics in screening, whether that be early detection or indeed in surveillance. And then in terms of the way that we're looking to use that, it's interesting that when I came to kick off the UMCCR, and there was a number of groups that we had, and then we probably have early stages of recruiting, it became very clear that there's a strong focus around tackling the cancers that are most challenging to our community. And the graph I'm showing here, I know that when you start some of these talks, you're never meant to start with a graph, but I just can't help myself with this because this is actually, this information has been motivating many of the lab's focus for some time now. And that is a summary here of while cancer survival has almost doubled in the last 30 years, it really comes at a cost. That there are some cancer types that have really been left behind. There are others that are fantastic success stories. But if we have a look, you can see in this graph here, which is showing you the change of 5-year survival as a percentage over the last 30 years, and this is Australian data. We can see some cancers up in the top left-hand corner where survival is still less than 10%. And we've not seen any real progress in recent decades, where others, the likes of prostate cancer, we've seen really quite remarkable improvements in the survival for that disease. So why are we seeing cancer stuck up in that left-hand corner? What are the sort of features that we see in those? We label these as cancers of unmet need. And the root causes for that poor survival and their recalcitrance when it comes to trying to improve outcomes are many-fold. And indeed, we've done some time thinking about how best do we identify what's driving their recalcitrance and then think about how we can tackle it. So the first one is some of these cancers are clearly quite aggressive, and they just don't permit you the opportunity to do lots of lines of therapy. We can't build up large numbers of patients and work them through large numbers of clinical trials like we might do for some other cancers, and that then becomes a problem. A second one is that some of these cancers, pancreatic cancer being a prime example, as we've seen a variety of targeted therapies or approaches come out, they've just pretty much ignored those approaches. So traditional trials going in and looking at pancreatic cancer hasn't been able to find decent segments which could benefit from those new advances. And then the third one, of course, is rare cancers. If a cancer is very, very rare and we don't see it very often, how do you build up the numbers to use a traditional, robust clinical trial approach to work out whether you should be using drug A or drug B or drug C? You just don't have the playbook to actually work with those. And then indeed, if we go through a little further, we have others which we call diagnostic conundrums. So these are the cancers whereby they're difficult to diagnose, they may be ambiguous to the pathologist, and if you can't even work out where that tumour comes from, and you can't understand the tissue of origin, then that actually becomes difficult to slot it into a traditional care path, let alone think about how else you might tackle it. We have to recognise that even across this entire spectrum of cancers, when cancers are detected late, we know that outcomes can be quite poor. And I also want to highlight here that while a graph gives you population information for the country, the reality is, certainly for the clinicians here in the room, there will always be patients that are outliers, that when you look down the microscope, they may look like they're a relatively benign disease, and you may be thinking about curative intent, but that cancer has completely different plans, and you can actually be a far more aggressive disease. And we think these are the key examples where we think that genomics can really make an impact on improving how best to tackle those issues. So in terms of how do you start to structure tackling those sorts of cancer patients, we've thought about that for some time now as needing core foundations, so technologies that we think we can plug in, that we can build to reach that ultimate goal of precision medicine. And these sorts of paths or circles or spirals, there's many different versions of this diagram that you'll see, whether it be working in cancer or cardiovascular disease or other sorts of illness. I think it's really the logic path that we've seen for the likes of precision medicine. And what I'd like to do now is actually walk you through showing you how we've been building some of those tools and then show you how we're actually starting to use some of those to tackle specific clinical questions. So the first step on this path towards precision medicine involves the building of large cancer atlases. The idea of integrated genomic analyses of large cancer cohorts have been absolutely key in giving us the understanding of what we believe to be the root causes of cancers, what we believe to be the driver mutations that are responsible for each cancer type, what actually, out of all those driver mutations, which ones may be druggable, which ones may cause a phenotype that we can exploit therapeutically with drugs that are currently available. And then we need to go forward and actually build a platform where in the pathology service, we're now looking at how we fully integrate genomic information into that early diagnostic component of looking at the patient samples. The next step I've got there is called ex vivo screening, and this is an idea that certainly in research settings we've seen for many years. We've learned a great deal by being able to take patient material, taking a tumour from a patient, be able to grow that within a mouse model, and then expand that tumour, and then be able to treat that tumour with a variety of drugs, giving us lessons to understand how that tumour will respond to a whole spectrum of drugs. And we can test more drugs than you ever could within the patient. So it's a way of being able to scale up that step, which relates to genotype and response to drug. And then finally, once we have that, we need to get much smarter about what are the clinical studies that we can enable. And indeed, this is a perfect place to do that. And then this is also a great way to on-ramp new therapies, either looking at vulnerabilities that are discovered by this approach, or new drugs as they come on. We can look for a biomarker based on these foundations to then actually drive those into the standard of care. All right, so if I start to walk through these one at a time, the idea of building atlases, well, anyone who's been subjected to one of my talks over the last decade would have seen this slide, and that is that all the work that we are doing here at the UMCCR is based very, very heavily on a set of atlases that have been built over the last decade by the likes of ICGC and TCGA. There have been very large international efforts where we have painstakingly collected patient material, collected clinical data. We have sequenced those tumours, and then we've tried to understand what is the actual root causes of those tumours one at a time. I guess one of the really exciting things about this is, as it's been such a large effort now, we have atlases that are starting to give us insights into every major cancer that is of global significance because of these efforts. Most importantly, the data that has been collected in an ethically sensitive and in a responsible fashion, so that the data can be made available to both academics and to industry to really accelerate our discoveries in cancer and really push forward to see how we can make precision oncology a reality. And with the appropriate ethical clearances, every single person in this room can gain access to the patient sample data that has been generated, whether it be genomic or the clinical data for those patients. This really has become foundational to so much of the cancer work we see here today. I should highlight that this has been a truly international effort. Australia has punched well above its weight in a number of programs involving many people from across Australia. I had the honour of leading that activity, which has looked at the likes of pancreatic cancer, melanoma, high-grade serous ovarian, amongst others. And this is data now that I think we take for granted, but has certainly been made available to the community to see how we can make the very most of it. I guess the second step about how do we build a census of actionable mutations, this is by now with that sort of data, how do we mine out which drugs which are currently available might work in any given cancer type. So here we're asking the question, if we have a drug that targets a mutation that was originally developed for breast cancer, how frequently does that Achilles heel occur in a pancreatic cancer or a colorectal cancer or a very rare brain cancer? And what we've certainly learned from these activities is that many of the pathways that are involved in promoting cancer when mutations are required are common across many cancer types. So this has really been a very a key component of ICGC that we've actually put all that sort of information together. Now, similar to before, like I showed you where that raw data is there for you to exploit, we actually have large amounts of clinical data now being integrated and laid on top of ICGC data. So when there are mutations in different cancer types, for all the drugs that are currently available, this has been collated and is available for anybody who wants to actually pursue that. And then finally, there's been some very recent work now which has focused on a large international effort to harmonise whole genome and transcriptome data to see how that as a thorough molecular tool can give us maximum insights into cancer diagnostics and the potential druggable targets that might exist in about 21 different cancer types. All right, so all of those ideas are at your disposal. They are readily searchable with the appropriate controls, and they're enabling a great deal of research globally. If you think about the tools that we've put together to do that research, that is actually a prime set of skills that we've had in a research setting. The challenge has now been, how do we bring that forward and integrate that into the likes of pathology? So how do we actually ruggedize that, scale that up, and integrate that into pathology services? And really, if you think about, once again, if you see anyone who's presenting on precision oncology across the planet at the moment, you'll end up with this classical sort of image. And we think about that we would take the likes of a patient in a precision oncology format, whereby that patient would present at a clinic. We would see that there is a sample taken from that patient. That will then go into a laboratory where the genomic pathology takes place. And there's a number of different tools that people will look to use, whether it be just looking at the DNA panel, and I'll touch on that in a moment, or whether we try to completely decode the genetic blueprint of that individual's tumour. And then we actually try to crunch that through the computers to come up with insights that we think may be useful to the clinical teams. And then finally, that information is passed on to an expert board, which will evaluate that information, integrate it with the clinical information about that individual case, and then think about how they're actually going to take that forward. The green box we can see there is one that we've worked on very hard to build tools to work on the likes of the whole genome and transcriptome side of that workflow. Indeed, here in the VCCC, we've been very lucky to be very close collaborators with Peter Mac Pathology, working within the researchers and indeed the services there in pathology services, which are already working on targeted sequencing. So we've actually looked to see how we can integrate into those processes and see how we can complement their activities where they would use a panel, and we can actually give a deep dive for those cancers of unmet need. This idea of a panel versus whole genome is one that we've all given lots of thought, and indeed it's one of those things that sometimes the scientific discussion, sometimes the theological discussion, it's always a financial discussion, but I hope that in the not too distant future, the cost of sequencing should get cheap enough, and the likes of cloud computing should make it amenable that we should be looking at very thorough testing for any challenging case. And I've just highlighted here, out of the two tools that we routinely use here now, how do we actually make the distinction between jumping and thinking about a panel versus the likes of whole genome and transcriptome analysis? And it's quite clear that panels are very well suited for looking at well-known cancers, looking at cancers which are primarily driven by a point mutation or well-known fusions, samples whereby you can only get a limited amount of material, or they may be very low tumour cellularity and you may need to enrich those cells. Or indeed, you have a drug that's approved that if there is a specific mutation, you can give those drugs. Those are the sorts of contexts where a panel is fantastic. And indeed, they overcome some of the deficiencies we have currently with the whole genome approach. For us, we think that whole genome sequencing should be used in the likes of cancers where the histology is ambiguous or they're a diagnostic conundrum. We would like to use it in cancers where we believe that point mutation isn't the primary thing that goes wrong within those cells to drive that cancer. And we know that there are many cancers where point mutation is not the primary driver. There are other rearrangements and things that can take place. We also know that when cancers evolve and when patients relapse and they've acquired resistance to many drugs, the cause of that can be either a complex genomic and or epigenomic event, and we'd like to be able to pursue those and recognise those in patients. And then finally, there are many cases where understanding the prognosis or the expected path of where your patient is going to walk, whether they're going to have a good outcome or a bad outcome, is absolutely key. And indeed, in the hematological setting, we're seeing for blood-based cancers that whole genome transcriptome can be very helpful with those challenging cases. All right, so if we think about the whole box and dice that we throw at whole genome transcriptome, and we think about what we learnt primarily from the likes of ICGC, we realise that we don't just come up with a gene list saying these are the genes that we think are likely to be of interest in this patient. We actually can extract and wring out an awful lot of information, the majority of which we think can have clinical potential. And I'd like to walk you through some of those right now. It is a very confusing diagram here. Suffice to say, this shows you the types of things we can get from DNA in light blue, the insights from RNA that are in purple, and then the funky one in the middle is where we actually put the RNA information and the DNA information together to understand more about the cancer genes in that particular patient. All right, so the first step really relates to this idea of if you have the normal DNA from a patient and then DNA from their tumour, we can very quickly decode their normal genetic blueprint, understand whether there is an obvious cancer predisposition in that family, and then we're also able to go and accrue or collate all of the mutations that have accrued in that tumour somatically. So these are the things that have been collected by that tumour as it's progressed over its lifetime. And those things can be the likes of SNVs or our single nucleotide variations through to copy number variations, the CNVs, or structural variations, so chromosomal rearrangements. And indeed, we need to be able to capture all of those things from the genome information to help make sense of challenging cases. We also are able to dig in now that we know that more and more cancers actually can be driven by the likes of pathogens or viruses, for instance, in head and neck cancer and in gynecological cancers. So we need to be able to go in and actively detect those as well, understand what was there and where it has been located in the genome. In terms of the way that we summarize that data and to give you an idea about what an unholy mess a solid tumor can look like, I'm just showing you here an example of the way that we summarize information for a whole genome. So the circle corresponds to, have I got a pointer? Maybe I can do it here. Then I'm not biasing the left versus right-hand side. This cartoon around the outside is showing you the chromosomes going from head to tail, chromosome 1, 2, 3, 4, 5, 6. It's known as a circles plot. And then when it's like an onion that we have a number of layers, this one here is showing you the types of somatic mutations that we see and the relative frequency of those. We're able to use this green color here is showing you where there's been regions of gain along those chromosomes. Orange is regions of loss. And then indeed, these little rearrangements and lines are showing you where the chromosomes, like your computer hard drive, have been shredded and have been rearranged and chunks have been deleted. We now have the ability to decode that rapidly in days for any given patient that presents here at the VCCC. The second thing we do is once you have all that information, what else can we extract from it? Well, we can work out whether that tumour was coming from cells that are diploid, so they have 2 copies of every chromosome, whether there's been genome doublings. We're able to get what is known as ploidy. It's something that we've used traditionally from karyotyping of tumours, but it's a way that we can estimate the overall copy number state, or how many copies of the genome might be within those tumours. We can estimate whether there's 1 or 2 or multiple clones. So are there multiple different types of cancer competing in that patient? We're able to estimate the mutational burden. This is very important, that if cancers are chock-a-block full of mutations, quite often the immune system with the right promotion may well be able to pursue those mutations and use that as a therapeutic option. We also have a number of tools here. MSI and HRD relate to looking for defects in DNA repair. If we find those specific defects within the genomic data, there are actually clear lines of therapy that we can use. And then indeed, there are other sorts of assays that we get on the side about the overall state of the genome, how robust it is, and indeed, are the likes of telomeres being engaged in promoting survival of that cancer. These tools have all been widely tested originally in ICGC, and now we've been working forward to see how we can use those as tools within the clinic. The next thing we want to do is, once you've actually worked out that additional information, we need to start to layer some insights on top of that. So now you've got all these events, you've understand the blueprint. How do we start to put those into the context of what's driving this disease? And then ultimately, how do we start to work out what's going to be clinically useful for that individual? And so we walk through a number of different processes there. Once again, these are codified, we go through and take that genomic information. We can identify what with confidence is responsible for making that malignancy grow. We're then able to overlay onto that which of those mutations may be useful when it comes to therapy. And we do that either using open source approaches or indeed in collaboration with industry. And I'll draw attention here to a collaboration we have with Purion-DX, which is a company that is focused on doing clinical prioritization and trial matching with genomic data. So in this instance, we decode a patient, we summarize the variants that we think are affecting the genes in that patient. We can then put that through our open source tools, but we can also then put that into the likes of Purion-DX, and then we can overlay onto that the insights that have been gained for that. Why are we actually taking that pursuit rather than just focusing on the likes of the open source approach, where we do know that people over time are putting in their information and there are plenty of insights to be gained? Well, it's simply because the likes of Purion-DX are tools that are now being adopted by large numbers of cancer services and pathology services, where they are collectively annotating variants when they see in patients, and those sorts of insights are now available to other people that join that collective. So as we use this now on patients, we can review what we've seen about a given mutation, but we can also rely on the knowledge base that's been collated by others. And I think in terms of precision oncology, we're really going to see some of the biggest changes in the next 3 years, where curation will become much, much simpler because of the sheer scale of information that's being collated and shared, so that we can actually recognize where the insights might be. All right, so the next bit we have there is RNA. So RNA is the molecule that we see being made by cells. It's the transient copy of a gene that's generated when that gene is active. We can use that RNA information to give us insights into the DNA mutations that we've seen, and give us confidence that when we find something and we think it might be important, we can actually be confident that it's doing the thing that we think it's doing in the cells. So just to highlight, we're able to, whenever we see a mutation, in this case you can see on this thing on the top right-hand side, the tumor suppressor gene called CDKN2A. And this is just showing a graphical example of a gene that we can see is being deleted in a patient's tumor sample. And what we're able to do there is actually have a look at where the relative level of expression in that individual would sit compared to hundreds of other cancers that actually don't have that tumor deleted. So once again, we rely on the atlases, and then we overlay our patient now to work out that we're confident now that this deletion is inactivating. We can use these sorts of approaches as well to make sense of when we see things that affect splicing, and we can do things where we see the likes of fusions, oncogenic fusions that can drive those things. So by having those two outputs, RNA and DNA together, and computationally we can overlay them, we can very quickly hone in on the areas that we think are likely to be of key importance to that individual. Neoantigen load. This is a term that relates to when there are mutations that occur in genes and those mutations are expressed and they cause changes to proteins. You have the potential of your immune system is able to recognize those peptide changes. And indeed, if there are particular events there which are causing potent neoantigens or large numbers of neoantigens, we know that those are playing a role in your immune response to cancer. And indeed, this is a very keen area of research, trying to understand how we can better understand neoantigen load and how that may make someone more likely versus not responding to immunotherapy. We should say that from, once again, the atlases that we've used, we know that neoantigen load can influence prognostically, and indeed is clearly playing a role, and even in things as challenging as here in pancreatic cancer, that tumours that have a high neoantigen load and high quality neoantigens, those patients tend to do much better than those that not, even for something as dire as pancreatic cancer. Tissue of origin. This might sound like a simple concept, but for some cancers, it is not straightforward as to where that tumours come from when you look down the microscope. And indeed, for other cases, when people are very confident that it's come from a particular place in the, it may be a tumour from a particular site of origin, when we sequence it, the pattern of damage actually doesn't give you confidence that that tumour actually comes from that site. So for instance, if we sequence a pancreatic cancer patient and we have a biopsy of their tumour, when we sequence it, we can formally ask the question computationally, is that a pancreatic cancer, or is this a colorectal cancer that is actually metastasized to the pancreas? Or is it a melanoma that's metastasized to the pancreas? These are examples that we've seen recently within the laboratory. And then just to highlight, RNA and DNA together is a very potent tool to actually make sense of this idea of tissue of origin. The colourful computer graph you can see on the right-hand side, this is work I really want to acknowledge Richard Tothill for, who's a member of the centre, is really a colourful graph to highlight the relative pattern of, or the assignment of approximately 10,000 different tumours to particular tumour types using a sophisticated approach. And basically you can see on the top right-hand side, all the different tumour types give you different colours. You can see that there's a set of gene expression that can be informative of telling you about where those cancers come from. And then the very small dark star that we can see here is a tumour that, from a point of view of microscopy, was ambiguous, and they weren't sure where that metastatic tumour arose from. But indeed, when you have a look at its global expression pattern, you can see very clearly that it maps onto the breast cancer cohort. This sort of approach is now a tool that we're looking to codify in here and have this routinely used on any tumour, whether it's meant to be of known origin or one that is ambiguous. Pathogen detection, as I said before, looking for the likes of viruses is something that we can do now. And in any of the cancer types where we know that that is going to be a priority, we'll actively look for that. This is just showing an example of when we found HPV and where it's located. In this instance, the HPV, when it's been inserted into the genome, has led to the amplification of the enhancers around MYC. And this is really an example of how cancers can actually be a oncogenic signal or promote the growth of this individual's tumour, as well as putting in those viral genes that can interfere with cell cycle and immune response. And then finally, just taking the RNA information alone, we can actually read out that information to estimate what we think particular processes might be doing for an individual, compared back to our very large atlases of RNA expression. So the top graph is just showing you the relative expression levels for a whole series of immune marker genes. And you can see that overall, based on that distribution, this tumour is relatively, has a low immunogenicity. We're not seeing a high level expression of those genes, versus the one down below, we can look at competency in the presence of DNA repair genes, looking at the relative expression of those for that individual versus hundreds or thousands of other individuals. So these tools are constantly putting an individual in the context of the knowledge that we've had before. All right, so the final tool I want to touch on is this idea of ex vivo modelling. And as I mentioned before, this is, wouldn't it be great to be able to take a biopsy of a patient's tumour and be able to grow that and screen that with all the drugs that we have available, and try to test sensitivity resistance profiles for that individual sample, and then return that information back to the clinic for consideration when you're actually treating that particular patient. And the concept can be done in a number of ways. I said originally people used to do it using mice. They would take these biopsies, put them into an immunologically challenged mice and grow them. The reality is that when we did that for pancreatic cancer, the cancer is so aggressive, by the time we got to the point of getting information from our organoids, from our xenografts, the patients had passed. So there was the lag was too great. Even though we could actually find that for different patients, there were exquisite sensitivities that we could test in those models. It was great for building the body of knowledge, but it was not suitable for prime time in a clinical setting. So this graph here is just showing on the top panel is showing a tumour that is genomically stable. You can see the circles there with not much damage. Whether that tumour was given gemcitabine or cisplatin, it would just grow. The graph is showing you the volume and the speed at which the tumour grows. This particular patient was given gemcitabine after surgery and had no response, and it matches what we see here. The bottom one is showing you a patient that, once again, after surgery went on gemcitabine and did not respond. The volume here is showing you, the graph is showing the information from the mouse models. But there actually is a drug there that we found that patient to be exquisitely sensitive to. And indeed, the genomic information suggested that we should be trying that drug, because in this particular case, the pathway involved in DNA damage repair was ruined. RPA1 in this case had a splice mutation that was causing it to be a hypermorph. This has been great for studying mechanisms of DNA damage and sensitivity, but the synchronization and getting the information for a patient, we just weren't smart enough or quick enough to do it back then. If you fast forward to where we are now, there is a great deal of interest across this precinct in looking at how we can take a biopsy from a patient and we can start to culture those tumour cells and be able to scale those up and do drug screening, either for known drugs or novel agents, and build up a whole new set of information around what mutations may be influencing druggability. I've highlighted here Fred Holland. He actually heads the UMCCR's organoid core facility, but he works very closely with colleagues here in Peter Mac. And indeed, I want to draw attention here to both Tracy Podolsky and the WeHi team that we've been working closely on when it comes to pancreatic cancer. And in terms of thinking about how readily is this for prime time, there's actually been some work only published last month showing now that quite impressively for the likes of metastatic colorectal cancer, these sorts of models are allowing people to screen and look at druggability in response to known agents and showing very good concordance between what's seen in patients, what's seen in the organoids, and indeed organoids predicting sensitivity within those patients when taken forward. We think there's a lot of opportunity in this and something that we want to be running side by side with our other activities. All right, so if that's the foundations that we've built, that's the sort of toolkit we really want to focus on in this walking towards precision oncology. I'd now like to take you through and give you some examples of how we've been starting to use this prospectively here in a variety of activities involving many of the folks here in this room. The first one relates to pancreatic cancer. You remember it was that cancer of unmet need that was in the top left-hand corner. This is a cancer that we know all too well that outcomes have been dire. The 5-year survival is still under 10%. We know that its incidence is increasing in the Western world, and there are predictions it'll become the second most common cause of cancer death in the US mid-next decade. We've done an awful lot of atlas work, and we know that if you go through, and we've now sequenced about 1,000 patients into our knowledge base now, which is by far the biggest knowledge base we have of this disease worldwide. There's about 70 genes that can all, in some way or another, damage a key pathway. So the trivial pursuit picture you can see here, each piece of pie relates to a pathway that has a gene that we've got evidence that can be damaged and actually contribute to the growth of these cancers. And this has been great in giving us the biology of what's under the hood of pancreatic cancer. The big challenge then is, well, how many of those things can we exploit therapeutically? And surprisingly, even though this is a cancer of very poor outcome at the moment, there are a whole pile of potential therapeutic opportunities within that cancer based on the types of mutations that we're seeing there. The challenge for us is that these mutations don't happen in isolation. We see that these are combinatoric mutations within our patients, and there are other layers there that we are yet to understand. So we need to be able to collect a large number of patients and prospectively do those sort of ex vivo tests, as well as seeing if we can use this information to kick off interesting trials for patients that are being diagnosed here right now. Just summarising what we've seen from the prospective work carried out recently in UMCCR into pancreatic samples. These patients are coming from the Australian Pancreatic Genome Initiative, from the Purple Registry here based in Melbourne, led by Belinda Lee, and indeed by Dan Crowe over at Monash, whereby we've been able to identify, surprisingly, a set of patients there that we think are could potentially be seeing treatments right now before we get too experimental. So the first thing I'd like to highlight is about 2% of those patients have been hypermutated. That means they contain thousands of mutations, they have a high mutational burden, they have lots of neoantigens, and they are prime candidates for the likes of immunotherapies. We have 9% of patients that are BRCA deficient. So you can see by the spirograph there in the middle, the circles plot, there's lots of damage to those genomes, and that genomic instability can be exploited by a variety of drugs. The ones next to that, the KRAS wild types, these are tumours that lack a potent oncogene, so a gene that drives pancreatic cancer in the majority of cases. One of the challenges for us is currently we don't have a drug that will combat that particular oncogene, but when it's missing, what we actually regularly see is a variety of mutations that are readily druggable, that there are drugs available that can be used from other cancers now to target those particular weaknesses. And then finally, as I talked before about the likes of tissue of origin, we've seen about 4% of our patients that present with a mass in their pancreas and has been presumptive to be pancreatic cancer. Indeed, once we've got in and we've been doing a deeper analysis of those, we actually find that the genetics is telling us that this cancer hasn't come, it's not a primary tumour, it's likely to be a metastasis from somewhere else. And then finally, there's about 30% of the patients that have one of these potential targets that we would really like to try to target using that ex vivo setting. That work is still to come. Just highlighting that in terms of even in something as deadly as pancreatic cancer, there is more and more evidence now coming out that if you can find that right genotype, and here I'm referring to the BRCA deficient phenotype, you can get exceptional responses in patients. So the one at the top was one from ICGC who had advanced metastatic disease that after one round of a DNA damaging agent, when they were BRCA deficient, lived for many years, ultimately dying about 4 years later from, but not from pancreatic cancer. And indeed others where PARP inhibitors have been very, very successful exploiting this vulnerability. We need to be finding this vulnerability in every single case that walks through the door. I should also say that the first good news that we've seen in some time now is the likes of PARP inhibitors doing good work in pancreatic cancer. It only targets those that are BRCA deficient. We're very keen to see how we can pursue that within this community going forward. Finally, the rare cancer types, in terms of looking at the ones that are KRAS wild type, there is now more and more data coming out that these particular fusions and events can be exploited therapeutically. The bottom two are looking at a pancreatic setting. The top two are well-known fusions from other cancer types where there are drug regimes that one can use. And indeed, we're now seeing the exact same drivers within those rare pancreatic cancers. Finally, that's interesting. We've got a bit of a font thing there. This is just to remind me that we do get organoid information from some of our patients that we've been steadily scaling up. And there are some instances where we have found exquisite sensitivity to particular agents, and we have been in our ethical framework be able to feed that back to clinicians for them to consider going forward. So you can see it's an iterative, stepwise approach. The second example I'd like to give you is cancer of unknown primary. This is a classical diagnostic conundrum. These are the cancers which are ambiguous when they go through classical pathology. It's unclear to know. It's clearly metastatic, but it's unclear to see where the primary can't be found, and it's unclear to work out what the site of origin was originally. The outcomes for these patients are very, very poor, and we've been very keen to see how we can enable the likes of Richard Tothill and his colleagues in an effort led by Linda Malishkin to see how genomics, whole genome and transcriptome can enable their activities. And effectively what those sorts of studies do, this is just showing you a case study of a patient that presents with two small metastatic lesions. The classical histopathology showed that it was a moderately differentiated adenocarcinoma of uncertain origin. The markers weren't consistent with a particular tissue of origin. But when we go through and do the genomic sequencing, we immediately see a set of mutations that are consistent with the likes of lung cancer. And indeed, the bottom right-hand side is showing you that the pattern of global somatic damage, the scarring across the DNA of the tumor, occurs at nucleotides and gives a pattern that is very distinctive of the pattern of damage that is caused by the carcinogen, which is in tobacco smoke. And indeed, this patient was a heavy smoker, and this is very clear indication that the tumor that we were seeing, despite its locations, actually was derived from a malignancy in the lung. A second area that we've been working, in this case with Peter Mack, the haematology service, is to think about this idea of the prognostic conundrum. Now, the colourful graphs you can see there are a number of different molecular subtypes. So if we classify cancer patients into a whole pile of different subgroups based on them carrying a particular mutation, there's been fantastic work done to show that those different mutations can predict that a patient is going to have either a very good or a poor outcome. And when you think about the likes of leukemias, you have a number of options that are available to you, ranging from the traditional chemotherapies through to transplants and CAR-T, and these things get increasingly expensive as you go. It's absolutely key with challenging cases when you can't estimate prognostically where they're going to go. Also, with any of these sort of cases, what you really want to do is have the most accurate prognosis of that patient that's possible, especially given the types of tools that are available to combat that disease. And so routinely what we're doing here in the likes of B-ALL, where these are the sorts of subtypes that we can see, and those particular subtypes can be driven by a wide variety of genomic events, we're routinely now using the genome transcriptome type work to come out and give a readout on some of these cases. This is a recent 17-year-old male that presented with BALL, and indeed, when you look at the readout on the bottom right-hand side, you can see that they have an IGH DUX4 fusion that was actively expressed, and indeed, these sorts of events can be, we can see on the left-hand side, actually have a very good outcome. So this, in this case, is a good news story for this particular individual. Finally, another example of one of the projects we've been working up is this idea of rare cancers. As I said before, rare cancers are challenging to treat because we don't have a playbook. There are many, many different types of rare cancers. They make up a significant portion of cancer-related deaths every year. And we can't do standard sorts of tools, clinical trials to make sense of those. And indeed, some of the activities we've been doing with Claire Scott across her Stafford Fox Rare Cancer Program has been now to drill down into rapid sequencing of challenging patients. And this has actually, in many cases, changed both diagnoses and care paths for patients. And I draw your attention to the one on the left, which was a diagnostic conundrum believed to be an atypical Ewing sarcoma. And the actual genomic event that took place, we could only identify using whole genome because there were 3 chromosomes coming together, and it broke the tie on some of the other path tests that have been done. And indeed, the sample that is second from the right, which is a rare leiomyosarcoma, in this particular case, this patient had advanced metastatic disease, and after the rapid turnaround of the genomic analysis, actually went from ECOG3 to, I believe they're back at work at the moment, because the PARP inhibitor was exquisitely successful. So we're actually building this information, but I think it's important to realise for rare cancers, we need to be sharing this data globally. And if we don't do that, we're really not going to be able to benefit these patients as well as we could. Finally, in terms of cancers that relapse, we know from the literature there are all sorts of mechanisms that can cause cancers to evolve, and they can develop an inbuilt resistance then to a wide variety of drugs. And luckily, genomics can be used to actually make sense of those events. And we've worked on that once again on our knowledge bases and our ICGC efforts. I draw attention here to work we did for high-grade serous ovarian, looking at how they might become resistant to standard DNA damaging agents. But indeed, we can now use that for patients by doing longitudinal studies, looking at their early tumours and looking then how they might come back. And then indeed, just showing some examples here, this is looking at 3 different patients here that we've identified, very different mechanisms that have caused their acquired resistance. One that was having radionuclide therapy, which has seen amplification of a number of oncogenes, another gaining a druggable mutation in the pancreatic net. And indeed, this patient here able to acquire both a resistance to cisplatin and immunotherapy after going through 2 lots of courses with a rare pancreatic endocrine tumour. So finally, with all those sorts of things going on and the knowledge that we've been building on those particular areas, we really look to see how we can take that to enable the community here. In early 2018, we kicked off the VCCC Precision Oncology Program, where we want to use this standard engine now, where we would look at using panels, or we would look at using whole genome transcriptome, and then see how we can use that to bring our community up to speed, and then start to see where we think the clinical utility may be in these approaches. I should say that the program has been done as a collaboration across the VCCC, and it has a large steering committee with champions located in every one of the key institutions. And indeed, these people have been instrumental in deciding when do we use panel versus whole genome, and how do we on-ramp patients into this particular study. Each of the tumour streams that we have here within the VCCC, we're invited to come together and build an algorithm of who do we think we should be using these sorts of approaches on, when should we use panel and when should we use whole genome, and that's been done by the clinical community rather than a boffin like myself sitting behind a computer, and we're actively pursuing that now. And then indeed, just to highlight that once that was constructed, we've gone through a lengthy process to align our genomic pipeline with the activities that occur in Peter-Mac pathology, so that we have continuity of that curation, but also that the reports that we start to generate will come out in a language that is compatible both with our colleagues that are in pathology, but also with the oncologists that will be receiving this. We don't want them to be getting 2 different reports in 2 different languages or dialects, which then makes it challenging for them. And really, we kicked off after doing a lot of training and compatibility in the earlier part of this year, and we're just under 40 patients now that have been recruited, either going into whole genome transcriptome or to comprehensive panel. So just in closing, I think right now what we tend to be thinking about with the world is we have lots of people working on clinical care based on what we currently know about prior experience, and we use traditional approaches. We have groups that work in the likes of clinical research where we're testing mechanism and looking for new opportunities. I think really what we would like to see is some sort of connection between those, and we think that the genomics and bioinformatics approach should allow us to accelerate how we move from the likes of clinical care and collate this genomic information through knowledge bases and smart technology to see how we can improve those patient outcomes. And with that, in the interest of time, I know we started a little late. I just want to take a few moments to acknowledge a large group of people here that have really made this exciting work possible over the last couple of years. Oliver Hoffman leading the informatic team, Kim Pham leading the genomics team. I want to acknowledge the likes of Linda Lee and the Purple Registry, which has been fantastic for pancreatic cancer, Claire Scott and Tony for access to the Stafford Fox activities, and indeed Jayesh and other colleagues for the VCCC program. Thank you.
Thank you, Sean, for that really wonderful and informative talk, really comprehensive overlook of what's going on in the VCCC. I'd like to open the floor up to any questions now, and I'll repeat the questions for the audience, maybe online.
Thanks, Sean. You gave a vision of linking the genomics in with functional assays such as organoids, and pointed out that in pancreatic cancer, the patient-derived xenografts are a real challenge because of the short survival of patients. Do you see any future for patient-derived xenografts, or do you think it's just not a tractable technology that can, bring functional information to the genome?
So I think xenografts are a fantastic tool. I think that you can get a lot of quality information from those, more so than you can get from organoids. But certainly in the pancreatic setting, when the work that we did, they were all primary resected patients. So that's the 15% where the tumour is so small, you've got a chance of trying to cure with surgery. And indeed, I think approximately 80 were generated. I'm talking here about work done by Marina Pajic in collaboration up in the Garvan. And I think 2 of those patients where xenografts were derived, the patient was alive still by the time the organoids are expanded to start doing the standard of care drugs, let alone something experimental. So there is they're a great quality research tool, but in pancreatic cancer, it's just too aggressive. I should also point out that in pancreatic cancer, xenografts grow in months. But as you saw from that survival plot, it's just not enough. So the organoids do fit. They're fit for purpose in that respect. And it's quite clear that organoids have a lot of potential in GI-related cancers, but I would really put a shout out to some of the activities we've been seeing here in Peter Mac, and indeed Richard's work around cancer of the unknown primary, where they're growing organoids as well. They're getting them from a wide number of tissues.
So there's another question. So I would like to ask a question, actually, and perhaps not necessarily fair to you in regards to...
It's just going to make me nervous. Yes, go on.
I think one of the challenges, I think taking my researcher hat on, putting my clinician hat on, is saying, how do we disrupt the current processes for clinical trials to actually bring forward some of the drugs that we need to use for these sort of N=1 studies? Because, if we're really going to make improvements, particularly for this group of cancer of unmet need, it's, as you yourself said, we don't have a playbook for this. We're going to have to find individual drugs, individual patients. But, we can't set up the trial just for one process for one study, which after 2 years closes. It needs to be a complete change in the way we do things so that we can keep adding more and more drugs onto these patients. And I don't know, I'm sure this process, something that's continued and thought of, but where are we in that thought process of how we're going to get this set up? Sorry, that's a bit of an unfair question to you.
No, it's not, I don't think it's an unfair question, because what's the point of building all these fantastic toys and then saying to a patient, congratulations, you have a vulnerability, and not be able to source the drug? So as you can see, this work has been for the VCCC. We're moving a lot of our activities now into the VCCC vehicle. It makes it simpler to collect patients. It makes it easier to get material in. It's going through a standard pathology pipeline downstairs, and we make the split, which is what the clinicians are after. The next piece is, how do we have the drugs available? Now, we're certainly looking at which drugs are likely to be the best drugs to be having available here in trials. There is we are yet to properly synchronise those activities around phase 1, around N of 1, but that is certainly the next focus that JS, Stephen, Grant, I talk about what we need to do next. We're also very lucky here in this community that there are a number of strategic partnerships that do allow us to pursue things such as the BRCA deficiency. That's a phenomenon that we first reported with the Sanger group around the mutational signatures in BRCA deficiency back in 2013. We reported it in pancreatic cancer, then esophageal cancer, then neuroendocrine cancer, high-grade serous ovarian cancer. The list went on and on and on. It's a global vulnerability that we really need those drugs here available to do that, and that's really being worked on right now with the team here.
Well, I think if there's no further questions, I'd like to thank Sean again. Please put your hands together. Thanks, Sean, for that really wonderful talk.